don't forget, right, that gene expression is one of the most important things associated to cowpea and cowpea. So this means that your initial microRNA profile might actually drives what genes are you expressing.
Welcome to MeatsPad, a platform to share breakthrough knowledge that is accessible to the entire meat industry. Be ready to hear from meat specialists who will talk about numerous topics in meat science, including animal welfare, meat production, meat quality, and so much more.
This podcast is brought to you by the Niche Meat Processor Assistance Network, the National Provisioner, UltraSource, the new standard for innovation, and Dry Age Pro makes dry aging in-house, flexible, safe, and affordable.
Hello, meat folks. Welcome back to the MeatsPad podcast. Today we have an excellent episode talking about meat nutrition with a very special guest. I'm going to have Dr. Phil Bass introduce him. Welcome, everyone.
Man, I'm excited. I'm pumped up. I'm ready to go. Really excited to visit with our guest that's here. And if it's okay, I'd love to kick it off and introduce that.
Yeah, go for it.
All right. So everyone get ready. So we have Dr. Amilton De Mello from University of Nevada. And I have to be careful because you spent some time at University of Nebraska, right, with the amazing Dr. Calkins and the other folks that were there. But while you're at the University of Nevada now, we know that you've been doing continuing along the academic route and learning. And what we'd love to hear is, what is some of the research that you're working on? And specifically, Francisco has already set the stage. Tell us about some of the work that you're doing with how meat and nutrient content is important.
Well, first of all, thank you guys for having me here today. It's a pleasure to be talking to you. Dr. Bass is a person that I already know. His reputation, his previous work at NCBA, it's great to talk to you. And Francisco is a great guy, so I talked to you before too. Thank you very much for allowing me to be here. Well, so, I am, as you probably know, I'm the only meat science professor in Nevada for now. So we do have a position coming next year. But coming to here allow me to actually work in different areas. So most of times when you are a meat scientist in a university that has more professors, you usually pick one single direction and you work, or you're a muscle biologist, or you just work with the food safety, or you're just a quality guy with fresh meats, or you move to a direction that you are working with processing meats. So my, when I got here, it was just one person trying to actually implement a new research program. So then, as Dr. Bass said, I was, my Ph.D. was in meat and muscle biology, and I was advised by Dr. Calkins, who is a specialist in fresh meat. So I did have a very extensive background in fresh meats as well as microbiology, because my master's wasn't focused on microbiology. So when I got here, our department was actually a merged department with the nutrition folks. And, I was reintroduced back as to the nutrition side by interacting with these folks. So, and since I had a background in food microbiology and a background in fresh meat, so I continued to work in that, both areas. And I also added the nutrition side because I did have a lot of support and folks that I could interact with. So I have basically three major lines of research right now. One is food microbiology, and you guys are familiar with. Mm-hmm. The second one is the fresh meats that we continue doing, studying basically most of the other things that people like to see too, how we improve meat quality. And this novel study area that we call like nutrigenomics, right? So this is something that we're trying to understand right now, because we all meat scientists try to develop research that can be immediately applied towards the industry benefits or public health. And I think that's are the three major research lines that I'm going right now. So it's the food safety, the quality in general, and the nutrigenomics.
And so in the nutrigenomics and the omics topics are that's the hot thing in a lot of the different sciences right now. So can you digest that a little bit down? And I guess no pun intended, but let's digest that just a little bit. Tell us a little bit about the focus that you have in the nutrigenomics and maybe, as you mentioned, how that can benefit public health by consuming meat. Isn't that great?
Yeah, absolutely. So the truth is that everyone knows about the effects of macronutrients, right? So why are you looking for nutrigenomics, dude? That's a question, right? So we all know that protein causes this. We know the lipids cause this. We know the vitamins cause this. So this is macronutrients. Everyone knows what a triglyceride is gonna do to you, or omega-3 is gonna do to you, or the vitamins. Iron, zinc, B12, B6, we know the benefits are there. They are well studied. But few people understand that when you consume meat, we're also consuming pieces of protein and small sequences of nucleotides that can actually be absorbed by your intestinal tract through the epithelial cells. And they can actually interact with these epithelial cells that we also generate another response that's probably going to affect the microbiota, or these guys can also reach the systemic, your blood vessels, and actually go target one specific gene that might be related to obesity, cancer, diabetes.
Wow. Okay. All right. So I'm gonna back up just a little bit, because a lot of our listening audience is not in the full-on academic mode, and so I'm gonna try to digest a little bit what you said, and hopefully I'm getting it a little bit correct. So wrote down a lot of things: macronutrients, and of course that's, like you said, water, fats or lipids, protein, minerals, vitamins, those kinds of things that keep us alive, right? And then you mentioned the nucleotides, and just to remind folks, what are nucleotides? Well, deoxyribonucleic acid it's kind of the building blocks of DNA in some cases. And so you're talking about there's specific things in meat that can be absorbed through the gut or remain in the gut and actually help in both ways. Is that what you're saying?
That's 100% correct, Dr. Bates. Yes.
Yeah. So, I mean, what a cool thing. We already know that meat is just an amazing food. Not only does it taste good, but it's good for you, right? But now your lab is experiencing some additional, or researching some additional health beneficial factors that meat can just help that much more.
That's correct. Yeah, and the good thing is that we were looking at both effects, right? Because as a scientist, we always need to keep our feet on the ground and be open to see all these results,
right?
Even they can be good or not.
Right. Yeah.
So the truth is that, as a meat scientist and I'm a meat promoter. I'm a meater. I was born and raised in South American country that consumes meat more than a lot of people.
Yes, they do.
So I probably have meat in my meals every day. So, and I'm a huge meat fan.
Yeah.
But the long story short is, when I wanted to actually understand some individual impacts of the meat consumption on human health, especially because there is this association with cancer. Mm-hmm. And, like 4 or 5 years ago, the World Health Organization released a monography associating meat consumption to cancer. But guess what? You can't prove it. So It's not science-based. Although it's science-based, it's not, most of the times,
cost effect.
We do not have a great methodology to rely on. And there is this huge appeal for, hey, why you're eating meat? This thing is bad for you. And at the same time, we have the meat supporters that said, hey, you don't know what we're talking about. Right. So these guys have vitamins. These guys got minerals. They got protein. They have actually creatine that works in our muscle building. Right. They have carnosine that actually helps you muscle fatigue. And then you're not eating meat, so you're not having all the essential amino acids at the same time. And there's always this balance that we can't ignore as scientists.
Right.
And we need to come back and say, hey, take a look at that, right? So especially what you're talking about, you might be not knowing everything you're talking about. Just come up with conclusions that you probably are creating in your own head.
Right.
Absolutely.
Well, and I'm a total carnivore, and I know Francisco is as well. And I'm sure we both come from, groups of people who also put meat at the center of every meal. And so we agree with
especially you.
Yeah, absolutely.
You be there. You're the guy who's there.
And that's a follow-up question on that. I think it's important to know, because we have meat processors, we have also consumers that may be interested in knowing more about this denaturation of the nucleotides that you brought up. Have you guys ever looked at different cooking methods, different temperatures, and their impact on the overall intake? We're talking about nucleotides and possibly the denaturation of these. I'm very curious about this, and I hope you can tell us more about this and its overall impact on health.
Yeah, sure. And this was the initial question that I had like 3 years ago, right? When you're talking about DNA and RNA molecules, you know that RNA molecules are not super resistant to heat. And our first hypothesis, null hypothesis, that was, can this thing survive? Is these microRNAs are not going to survive after cooking? That was our initial null hypothesis tested. And the interesting thing, man, is that, we were extremely confident that microRNAs will not be available after cooking. Mm. So I worked with two more collaborators here. One is Dr. Brad Ferguson. He's from the nutrition side, and Dr. Moser Fonseca. And then we sit down and said, hey, these RNAs, they don't survive. That's why you collect RNAs right after the animal is dead, or you need to do some biopsy, because you're not going to be able to, they're not resistant. And guess what, right? So we realized that some of these microRNAs, they actually are overexpressed post-mortem for 14 days. What happened is you start with like a certain amount, and after the animal dies, right? So that microRNAs are overexpressed. Boom, boom, boom. They go into 14 days and boom, and come down. So that was the first discovery that I did say, hey, these guys probably are great biomarkers for something else. Uh-huh. Everything that happens post-mortem. So you have something growing right there. They're great biomarkers. Let's take a look at that in the other side. But since our focus was on nutrition, I said, okay, we have actually like twice more microRNAs after 14 days. So we actually increased that concentration of specific microRNAs, and we tested 9 of them. So, and then the second question is, but what happens if we digest, if we cook, and what happens if you digest? Because I want to know if these microRNAs are going to reach my gut and they're going to be available there to be absorbed. So we actually cook them 70 degrees, right? We use the same procedure that we always do.
70 degrees Celsius for the
Celsius. I forgot about it. 70 degrees Celsius. Yeah, come on, man.
About 161 degrees Fahrenheit. Very good. Yeah.
That's right. 70 degrees Celsius, right? And it's recommended by American Meat Science Association guidelines. And then for taste panel. And then we digested that in vitro. So, and the digested material was analyzed to see how much microRNA we have in that digested material. And we know, right, what, the science says that these guys are actually absorbed in femtograms, which is super minimal concentration. And when we analyzed our samples, digested samples, they came out in nanograms. It was 2 more units than we wanted. So theoretically, we have a significant amount of microRNAs that are they're reaching your guts after passing through the duodenum, and they are there. They are still available. Yeah.
And so they're not being so these microRNAs, so these tiny little fragments of protein stuff, right?
Right?
That so it's actually not it's not being broken down during cooking. it's not being broken down in our rather acidic stomachs. It's actually making its way all the way to our intestines so that it can be absorbed. That's what you're saying.
Yeah, one thing that we cannot forget, that's this a long chain, super long, right?
Yeah.
And these microRNAs can be generated by the degradation anyways.
Okay.
So the degradation of the dead DNA can actually generate some microRNAs, but that's not what we're going after, right? We strongly believe that the microRNAs there, they actually are protected because there is a specific protein called agoprotein that actually englobes that microRNA and protects that guy through all that process. Mm-hmm. So, and if you look at the microRNA biogenesis, right, which is something super fun, and I'm not going to be digging this right now, the rest of people are going to get crazy. I got that. But I mean, so what happened is when these microRNAs are actually going through their synthesis, so these guys are actually produced by a single messenger RNA, and there is a protein called AGO2 that actually holds that and protects that guy through a different process. And also, right, so when you're digesting, never forget that the lipids are there too. Mm-hmm. And the lipids can fats, actually, they can bypass by holding that protein. Mm-hmm. And you eventually have protein there that actually some kind of way protected.
Yeah. So isn't that fascinating? Biology is so cool.
Yeah, it is so cool.
A little break. We'll be right back.
The Niche Meat Processor Assistance Network, or NPAN, is a proud sponsor of the MeatsPad podcast.
Can you tell us so are there specific species that you're working with right now as far as the different meats?
We're working with beef right now.
With beef predominantly? You're beef guy. That's all right. I'm okay with that.
And the second one, we're going to be beef against plate-based.
Really?
We're going to have a lot of fun right there because we have micro-vegetable microRNAs too, right?
Okay, sure, yeah.
But yeah, we have all these species coming down the line. Yes, we have everything lined up. It's just a work that we are putting our heads. Like we started this like 2 years ago, and it's super novel. There are a lot of new stuff that we're looking after. We're having a lot of fun. That's the most important part.
Well, yeah, and that's, I mean, of course, science is fun. Science is cool for those who those naysayers that are out there. Well, are there ways that you can actually adjust the nutrient content in meat, either at the really precise level that you're talking about, or say more on a grand scale for folks that a lot of our listeners are packers, processors? What are some things that they might be able to look at and tell their customers ultimately on what they should be looking for as far as meat nutrient content? Yeah,
so I understand that this podcast is related to, let's talk about the industry a little bit. I think these guys are going to have a lot of fun, right?
Yeah.
So in my understanding, all the industries, not only the meat industry, right? So the industry works towards demand. So if you have a higher demand, right? If you have a higher demand for masks right now, right? Mm-hmm. Or for hand sanitizers, even Tesla is doing like hand sanitizers right now. So it's an opportunity for demands. So in my understanding is in the future, we will have more understanding about what people want. So today there's a huge concern about grass-fed and grain-fed local, right? I want to get some omega-3s. I would prefer to get grass-fed beef. But we know, meat scientists, we know that's 1 or 2% more.
Yeah.
So
you'd have to. So for those listening, the data says I think you'd have to eat about 14 pounds of to equate to get the same amount as you get out of a 3-ounce salmon, which I try to eat 14 pounds of beef. I do, but yeah, you're not going to get.
We're talking about
for other reasons, right?
and I have these discussions with one of my students, and I say, hey, this amount that we increase by adding by just using grass or other supplements that you increase like 1 or 2%. If you look at the Western diet, that's like 21 omega-6 omega-3 ratio right now. Mm. You're just adding this. It's not going to really affect anything. But guess what, right? The industry needs to be worried about that because that concern might actually lead people to consume something specifically, not related to science, but their own preference or their own beliefs, right? So, you're coming back to your question now. we don't know yet how much we can. We are actually running a trial right now. These animals are going to be harvested like probably a week and a half, where we're going to actually look at the microRNA profile of animals fed different diets.
Mm-hmm.
So there are some things in the literature that actually show the difference between animals fed different things, different feedstuffs. Some are than corn, some are than wheat, some are than grass. We had our own here, and you're going to be able to come up with microRNA profiles from animals fed different feedstuffs.
Really?
And then obviously, is we review a lot of things in there.
This is awesome, and I thank you, Dr. Amilton, to sharing all that information. But I think, following up on the last question from Dr. Baz, and then you bringing up the industry. Your hypothesis, what you found was that on those 14 days postmortem, you found that those microRNAs, they went up. So let me go back, and for those folks that are processing meat in those countries, South America and Mexico, Central America, we don't typically have what we call the wet aging process, because we discussed that with Dr. Bass when we talk about beef tenderness, but they might have, depending on the genetics, we might have some differences when we talk about proteolysis or the protein degradation, which
I think that aging it's not really intrinsically related to nutrition, I guess. The nutrition values are going to be there. I think that the aging side moves more to the quality thing. Which is something that Dr. Baz is actually probably studying right now, because I am partially familiar with his research. And then, what I feel is that the nutrition side of this is probably not related to the time spent post-mortem. And we know that meat has 20% of protein, 7 to 75% water. What really varies there, which might change your nutritional values, is your lipidomics, right? So come back to omics, right? It's your lipids. So, we know, I personally believe that lipidomics is a very interesting thing, but we know that fatty acids and fats and cholesterol and triglycerides are the most important components related to health associated to meat. And these guys vary from 1 to 10%. Unless you're eating a Wagyu that, is probably 25% of fat. But it's more associated to the fat content and not the, the time that's spent post-mortem. But guess what, right? So coming back to your question, and then you say, okay, why is this increase of microRNA is important, right, for the industry in general, or for these guys? So you guys remember that I mentioned that I'm feeding two types of feedstuff in animals, right? And we know, so here's how microRNAs work. MicroRNAs, they actually penetrate and go, they reach the blood system, and they go there and attach to a specific gene and turn that gene off. Don't forget, right, that gene expression is one of the most important things associated to calpain and calprostagnin. So this means that your initial microRNA profile might actually drives what genes are you expressing and you are suppressing. And these genes might be related to attributes that you wanted to know. And these are things that I cannot tell you yet.
Right.
That was probably
future episodes.
But this comes back to what you guys want to know related to the industry, right? Why this crazy research is more applied to the industry in general, not only to the public health. Because this nutrigenomics thing is all about public health, but what the industry has with it. So if you are able to identify what microRNAs are being expressed and overexpressed in postmortem, and you depend on the postmortem timeline to have specifically final attributes that you want, you need to know what microRNAs you want to have there.
Absolutely.
Yeah.
Great.
Just put a bug behind your ears.
Right. Yeah. Yeah. Well, and you bring up so many crazy, awesome, different directions that, we talk about the rabbit hole that we all kind of end up going down in the academics, and as we continue with research and that's it. I think that's important to stay open-minded about some of these really precise, what we call basic research, right? To understand the tiny little molecules that can ultimately affect a lot of things later on. And that's kind of the premise that I'm hearing here.
That's right.
There's a lot of different things that we still don't know about meat science, and we're learning.
And you see, Dr. Bass, going back to all the mitochondria behavior, right? All proteins that we need to give you proper color, right? Yep. All of these are mediated by genes.
Right. Yeah. And if you can turn them on or off.
If you can turn it on and off, you can make a mess or you can improve it, right?
well, let's hope we continue to improve on that. Let's lean in that direction, shall we? So.
Well, we believe we can improve.
I believe that too.
That's why we study that, right? To come up with solutions that can provide a betterment for all the industry and meat quality in general.
Yeah. Well, and I'd like to ask one follow-up question, and maybe that could kind of wrap the discussion up here. But we do have to continue to investigate, and we do have to continue to use different thoughts and different minds. And this kind of roundtable that we're having right here is a pretty cool example of how we have representatives from United States. We have a representative originally from Mexico. We have a representative originally from Brazil. And I'm gonna point at you, Dr. Amilton. Can you tell the audience just a little bit about your original background? And you got a D.V.M. And then you decided to come all the way up here to the United States and spend some time researching and continue on with a Ph.D. And that's absolutely incredible. And you also mentioned you came from Brazil, which definitely has a different culture and a very beef-centric culture, right?
Totally different culture.
Maybe just wrap the discussion up on what it is that maybe drives your interests, and maybe how that gives you kind of a different perspective to the U.S. and North American beef industry, and then all the others that you've experienced along the way.
Yeah, sure. I was born and raised in a small town, and always been involved in farm activities since I was young. And in order to, and when I was working on my D.V.M, when I was at school, in order to have a good job, a good position in the meat industry in Brazil, we need to be a veterinarian. Right? Or you can work and get your bachelor's in animal science there, and but if you were a veterinarian, right? So most of the veterinarians are, actually being absorbed by the industry. The meat, if there is an industry that absorbs veterinarians and pay them well, it's actually the meat industry in Brazil. Okay. So this is why I decided when I was in my fourth year vet school, I took meat science classes. And I said, well, I want to work with production. I want to be a veterinarian that works with production. So, and then after my internships were in two major big plants right there. And after two major big plants, I decided, I want to know more. So I went to the master's and got my master's at Sao Paulo State University. Was like a microbiology focused on. And at that time, I was reading a lot of papers. I got into science, right? So I got into science, and I had an opportunity to read a lot of things from American scientists, right? And I finished my master's. I started my Ph.D. in Brazil. After 2 years, I applied for a Fulbright scholarship. So, and Fulbright actually awarded me a 4-year scholarship to come to the U.S. and get my Ph.D. here. Yeah. So after this Fulbright, I needed to go back to Brazil. I stayed 2 years back. I came here, got my Ph.D., went back to Brazil for 2 years, and I was teaching in a vet school right there, like meat science courses. Yeah. And then after 2 years, I was hired by a company, so they brought me back. After 3.5 years, 3.5 years, 3 years and a half in that company, my desire for research and labs and crazy stuff that you keep doing, and don't stop dreaming, and don't sleep anymore. So it's time to go back to academia. So I ended up coming back to academia.
Yeah.
That's the story.
Well, and that's an awesome direction, and the Wolf Pack is, I'm sure, very happy to have you. So thank you so much for joining us.
Oh, thank you very much. And just something that I forgot about your question. I think that the beef industry in the United States is amazing. It's something that we cannot see anywhere. I've been in several countries before, right? The quality, meat quality, the consistency that we have in the U.S., you can't find anywhere. And then all of these is fantastic, and that's what probably drives me to keep looking for things that can make this better and better and better.
That's awesome. Thanks.
Well, thank you, Dr. Amilton, for your time again, for being on this podcast. It was fantastic having this conversation, dialoguing with you. We hope to see you in future episodes and having more roundtables with you. We appreciate it, and we'll see you the next time. Thank you a lot.
Oh, it's always a pleasure to be here. Thank you very much, Francisco.
Thank you.
Thank you, Dr. Bass.
All right.
Take care, guys. Bye.
With that, we end this episode. Thank you a lot for listening. If you'd like to receive notifications on the new releases and the new episodes, please subscribe at www.MeatsPad.com. If you're a small and mid-sized meat processor and you have concerns or questions about a certain topic related to meat science and meat processing, please email us at info@meatspad.com. Thank you, and see you the next time.